So I'm Andrea, and I'm a second-year PhD in the Max Planck Research Group for Behavioral Genomics in the MPI for Evolutionary Biology in Pleun. And I will be talking about my project, how migration might be informed by residents through their genomic variation in our migratory bird. So first, I would like to give you a quick definition of migration. And migration is the directed, coordinated, and periodic movement of either an individual or population. And here I'm showing you some examples. And you might not think I'm missing one of the most important ones, or this is not the one that I was thinking of when you said migration. Yes, because we have bird migration. And bird migration is an interesting. It has interested people for centuries by now, and you have bird species that either do not migrate at all, or you have bird species that are complete migratory, and you have, of course, those bird species that do a bit of a mix in between, and that's what we are studying in our group. So we use the perfect model species, the Eurasian blackcap, to study this, and I need to sort of say why we're studying this species with the idea of the genomics behind migration. The Eurasian blackcap flies during the night on their own. They don't have any influences of their parents or anyone else. So it's really also found from previous research, a genetic component is involved, and most likely we can put it out better in this species. So I'm doing this talk a teeny bit differently, as I'm sort of going to show you my results, very recent results on a new data set, and then explain why we are doing what we'll be doing in the future. So first, we have a whole genome sequencing of 189 individuals. They're all males. And we have a chromosome level reference genome, And our populations are all distributed around Europe and Asia, the north of Africa. And the blackcap is fascinating because it has a wide variety in their phenotypes. So I'm just going to show you what we have of each phenotype. So we have, for example, here long distance southeastern migrants. Here we have medium distance southeastern migrants. We also have here, unfortunately not on the map, Georgian southeastern migrants, medium distance. We have medium distance that fly to the southwest, so they fly to here, and the eastern fly to here. We also have a new phenotype which has been around for 70 years that flies actually to the northwest of England to overwinter. And we have migrants that fly in short southwestern distance. And then where sort of my project will come in mostly is in the residential populations. We have 3 continental populations on the Iberian Peninsula and Morocco. And then we have our island populations, which we have Mallorca, a very recent island residence from Crete. And here are 4 archipelagos of the Micronesian islands with Madeira, the Canary Islands, Azores, and Cape Verde. And I mapped all of these samples against our reference genome, and I called SNPs. And so now I'm going to talk about what we're seeing in population structure, and to sort of foundate why it's interesting to check out these resident populations and the genomics of them. So first, I'm going to show you some admixture plots some admixture analysis that I did. I need to explain one thing. So admixture, when you run it multiple times on the same K, it actually gives different outcomes. So one should run it multiple times and then analyze it with, for example, a different program. So I use 2 programs, Pong and Klumpek. I'm only going to show you here the results of Pong. If it is, I can pull out the results from Klumpek, but it's not good. They're very similar. It's just that Pong is a little bit more clearer to show what is happening. So I did sort of find out that for K's higher than 2, I needed to run 100 times admixture, because it wasn't giving me enough confidence of how many runs were foundation of the best run that I find. So that's why I go from 10 to 100 runs. So let me first talk about what we see. So here is the admixture of K2, and 10 out of 10 ones, they are exactly the same. They always show this. Also with the compact, it always shows the same thing, and it shows that we have separation along, separating of the Micronesian islands with some admixture here in Madeira and the Canary Islands, with everyone else from the continental European samples being in a cluster. If I go to the next K, we have K3, and we actually still see the differentiation with the Micronesian islands, but now we can also see that the continental European samples are differentiating alongside of migrants and residents. So the residents here are continent Crete and Majorca. We see that most of the migrants have a mixture of a resident and a migratory phenotype. But what happens if we delve further? So next K, we see still the same for the Micronesian islands. And now we actually see that the continent and short distance southwestern migrants, which are in the same geographic areas, because like the continent is Iberian Peninsula, the short distance southwest migrants are also in the Iberian Peninsula, and they show admixture from the continental samples, the residential continental samples. Furthermore, we can see this group popping out alongside with Majorca, and this group of migrants are actually our Georgian migrants. And we have recently sequenced these and added these to further inform our results. But for the rest, we can see that migrants are still together, and we see everything else the same. The last K that I'm going to show you is K5, and here we actually start seeing that Majorca, Madeira, and Canary Islands are forming a cluster, and the Georgian migratory samples are on their own. So I do have to say that the optimum K is 2, maybe actually only 1, but at least up until now we can sort of explain all of what we're seeing here based on geography and based on their phenotype. So what is happening if we check it out in another way to see population structure? So I also did a PCA. I forgot to say one thing. Sorry, I will continue in a bit. For this data set, I used 360,000 SNPs, and they are all filtered in such a way that linkage disequilibrium is almost cancelled out in that sense. So they should be all independent of each other. Okay, back to the population structure. So first, let me just start with the migrants. And the migrants, we can actually see that we have one giant blob of migrants, sorry. And they play out in alongside PC2, with these 2 clusters being Georgian samples and these 2 small clusters being the short distance southwestern migrants. This is an improvement on our last study, which just clumped all of the migrants together. But what happens if we add our next continental population that is our continental residents? They actually continue on along PC2 by clustering here. So we clearly see what we also saw in the admixture plot, that we have migrants clustering together except for Georgia, and we see that the continental residents are are also clustering separately from the migrants, but on the same PC, and we see the intermediate form here of the short distance migrants. Let's now add our other islands. And we first start with the Micronesian islands and to fill out PC1. So first we have the Madeiran samples, then we have the Canary Island samples, the Cape Verdean samples, and the Azores. And here you can clearly see that PC1 distinguishes the continental samples here, regardless of residency or migratory, or whether they are migrants, from the Micronesian islands. And also in sort of geographic distances, these were the closest by islands to the furthest away islands. However, we still have our two leftover islands, which are Mallorca here, nicely clustering above the continental residents, which postdoc previously found also that the Mallorca samples, that Mallorca individuals would fall out of the Iberian cluster of the black cap quicker. And our last but not least are our Crete samples, which are still sort of clustering here together with our migratory samples, which is also not unexpected as the Cretes are only resident since a few years, so nothing much genetically. We are not expecting a lot of genetic variation to be found there. So there are two things that we can take away from this, that Micronesian populations distinguish themselves clearly from the European samples. This might be just geographically, but it can also be a relation of them being residents. And we have continental samples separating based on migratory phenotype. So now you might wonder, okay, we see something in population structure, but what do we actually see something also in morphology? So I am showing you here migratory and resident bird. You might not clearly directly see the difference, but there are some small morphological differences. For example, bigger brains or shorter wings in residents compared to the migrants. Furthermore, we also know that previous research done in the 40 years ago by crossing individuals residential from Cape Verde with southern German samples, they resulted in an intermediate phenotype of the migratory migratory restlessness, which is our leeway to what is our way to sort of quantify the migratory phenotype. So these are the Cape Verdean samples, these are the South German samples, and these are the hybrids. So we know that there is a morphological component. We see there is structure happening. We know from previous research that one can actually breed a resident bird in a few generations. And from a previous data set that I had on our old reference and with a little bit less samples, I could already see that based on the pairwise FST here, for example, of the Canary Islands always compared to the other populations that I was using, that the genomic fingerprint is quite different along the genome. And now the idea is to use, the idea is to answer the question, how did the island residents and the continental residents evolved from the migrants? So we are gonna do further population genomic inferences, and for example with machine learning, sweep finding with machine learning and transposable elements checking out how the transposable elements differ between the residents and the migrants, all with the goal to understand what is happening in migration. So we want to open up our black box of residency, which is still sort of a black box because not many people have done anything with it, and any of our results or genes or genomic regions to inform our black box of migration. And we hope to have soon new future results, which are hopefully going to be very interesting. And I think I'm way too quick, but thank you all for listening, and I'm happy to answer any questions. And also thank you to my group and the collaborators and the IMPers. And that was it.